Hi, everyone. Thank you so much for inviting me. Um, yeah, I'm really excited to present our work today, not just because I'm always excited to present our work, but because I get to present work that we released just 2 hours ago. So I think, and I know AI and biology is probably new to most people here, but I saw some Genentech folks in the crowd, so I think even for you guys, this will still be new. Um, so we start really with this idea that life is programmable. Um, so many of the functions that your cells, the cells in the body perform, are performed by proteins. And so, what is a protein? A protein is a sequence of amino acids. There are 20 naturally occurring amino acids, and we can represent each amino acid using a character, which means that a protein is effectively a sequence of characters. That sequence then can fold into a particular 3D structure, and that structure will inform its function. So in this case, we're looking at the structure of the green fluorescent protein, which creates a chromophore, which then fluoresces light, and that's how you get green fluorescence in jellyfish. So the idea here is like if you can learn to read and write the language of proteins, then you could learn to modulate, modify, and really understand biology and create new biology. The structure and function of a protein is really embedded inside of its sequence. So the sequence determines what the structure and the function of the protein will be. Um, and so you can imagine that if you could have really strong representations of a protein sequence, then you should be able to pull the structure and function of the protein out of those sequences. And we have, scientists have sequenced billions and billions of protein sequences these days. Sequencing costs have dropped immensely, and the amount of protein sequence data available has really risen dramatically. And so, if we can apply some of the tools in modern AI to this type of sequence data, then maybe we can pull structure and function of proteins directly from their sequences. So, I'm going to tell you that we're going to apply language models to proteins, and you're all familiar with language models of text. So you can do autoregressive or mass language models, where you're next token prediction or filling in missing tokens. The same concepts can be applied to protein sequences. Instead of tokens, you're either predicting the next amino acid or filling in a series of missing amino acids. Our models, ESM models, are mostly mass language models where we're filling in missing amino acids in a sequence. And the core idea here is that if you can learn which amino acids are related to each other in a protein sequence, that will actually tell you a lot about how a protein is structured and what its function is. If a word is doing a lot of work to carry meaning in a sentence, then the model has to learn that meaning in order to model that sentence. In the same way, if an amino acid is like a key functional site for a protein, then the model will need to learn that amino acid is critical for the function in order to be able to model its sequence accurately. So today I'm presenting our new models, ESMC, which is a language model of protein sequences trained on 2.6 billion proteins. Um, and on top of that, ESM Fold 2, which is our latest structure prediction model, which can enable really high accuracy structure prediction and can also enable the design of novel proteins. So one of the things, when I say structure prediction, I think a lot of folks have heard that AlphaFold solves structure prediction. So what is the thing that we're really looking for? So what AlphaFold really went after is the prediction of individual protein structures. And that we've made a ton of progress on that problem. But one of the key problems, if you really want to think about like designing functional proteins, is the design of protein-protein interactions. How does a protein interact with other molecules? And that's a problem that's still really hard and is in particular hard for things that are challenging therapeutic modalities like antibodies. So something like 30% of modern therapeutics, modern drugs, are antibody-based therapeutics, where you're injecting someone with an antibody, and that antibody is targeting another protein or another molecule in your body. And so if we can really accurately predict the interaction of antibodies with other molecules, then we can design better drugs and design better therapeutics. So ESM Fold 2 gets state-of-the-art performance on both general protein-protein interaction, but in particular is really strong on antibody-antigen structure prediction. We really spend a lot of time thinking about how we can translate increased compute into increased performance. And so we find that one, we want to make ASM fold as fast as possible. Um, we spent a lot of time simplifying the architecture and also implementing kernels to make ESM fold to the fastest model, structure prediction model. But not only is the fastest structure prediction model, we can then use that speed to generate multiple samples from the model and then ask it to basically rank its own predictions. And as you draw more and more samples and ask it to rank more and more predictions, um, the model gets better and better. And not only does it get better, we actually find that smaller models, like ESM Fold 2 Fast that I'm showing here in blue, which has half as many layers as the larger full ESM Fold 2 model, eventually actually catches up in performance with the ESM Fold, with a larger model with enough seeds. And so using enough compute, we can have some, like really small models that achieve really, really high accuracy in this task. One of the core innovations of ESM Fold 2 is that we use a recurrent architecture based on some of the recent looped transformer work. Um, so one of the big challenges in structure prediction is that we really only have like 200,000 or so real structures of proteins. And then if you take that and then you look at, hey, a lot of people have deposited structures where two structures are really, really similar to each other. Maybe one is bound to a particular small molecule and one isn't. And so there's a lot of things that really aren't two totally independent data points. If you deduplicate the number of structures, there's maybe 20, 40,000 really unique structures of proteins that we have available in total. And so that presents a really challenging problem for modern AI because most of the techniques that we look at will overfit really easily on this data. So one of the things that we can do is we can take fix the number of parameters in the model and then feed the representations from the end of the model back into the beginning of the model. And then and it's kind of important to take that recurrent update and constrain it so that it's not so that it can't so it's contractive basically. So you're not going to get the activations increasing unbounded over the recurrence. And if you can do this, then you can actually backpropagate through that full loop. And we find that that really increases performance on structure prediction and particularly increases performance on antibody antigen structure prediction. So we want to eventually take these models and translate them into the real world. And we do that by designing binders to 5 different targets. These are targets that are relevant in cancer and other diseases. And we design them in 2 modalities. One of them is a mini binder design, which is generally easier. And that's in the top row. And in the bottom row, I'm showing the design of SCFVs, which are a really clinically relevant antibody derived modality, where we consistently get really, really high affinity, nanomolar, even picomolar affinity in some cases, in all modalities for all targets. And yeah. Another example of so on the left, you can see the hit rate across all modalities and targets, but on the right, you can also see how increasing the compute that we use to generate the targets in the lab or in silico on GPU can actually translate directly into increasing the hit rate in the lab. So what we do is on the smaller circles there and the smaller unfilled circles, show our hit rate when we're using a certain level of compute to design, possible candidate binders. Then the filled in circles above each line show what happens when we basically use more compute to generate more possible candidate binders, and then also take each binder and rank it using orthogonal versions of ESM2, basically as reward models to predict how likely is this to be a real binder. And the more reward models you use and the more candidates that you generate, the more likely the final ones that you select will translate into hits in the lab. So this gives us a path towards scaling digital compute to yield increased performance in the physical world. I'm not going to be able to touch on all of the interpretability work, but I want to say a few things about it. We also spend a lot of time digging into what the SMC is learning and what is it representing as it trains on these protein sequences. And we find a really interesting decomposition of biology, which touches on, residue level chemistry up to domains and like represents active sites in really interesting ways. I'm gonna show one example, which I think is illustrative. So there's this particular motif called a nucleophilic elbow, where a particular type of chemistry is really useful across a large space of biology, and because it's so useful, it's actually evolved convergently multiple times in unrelated proteins, and you can see, actually, we find that the ESMC uses a single feature, single SAE feature, to represent the nucleophilic elbow across evolutionarily unrelated proteins, which suggests that it's like has a understanding of this protein function that is shared across evolution. So, kind of bring this to a close, we've got we really think that this is a new paradigm for biology, where we're moving this from an art form to an engineering discipline, where we can have really accurate representations of biology, do experiments on chip, on GPU, and have those experiments translate directly into the real world with high success rates. And ultimately, this is our work that is really without a lot of feedback between AI and the laboratory yet. As we kind of expand in BioHub and are able to collect more data in the lab and are able to really expand on that side, I think that we'll see even greater results. Um, so I want to say thank you to all the team and at BioHub who helped with this work, and thanks very much for hearing my talk.